happy to chair this session, and our speaker is Lea Popovic from Concordia University. We also know each other over more than 10 years through conferences and through visits and so on. So today, Lea is talking about spatial effects in stochastic dynamics of intracellular networks.
Thank you. Thank you, Ruth and Jeremy, for putting together this very interesting workshop. For you to hear some actual biology and data in action. I live in a math department, and I was trained in the stats department. But my interest in this project was that I've seen. I had done some work on multi-scale results for chemical reaction networks, and I have seen many models for stochastic spatial results for ecology, for population genetics, but I was kind of curious what kind of questions and how the models would be different if one asked them in the context of intracellular chemical reactions. So the questions are very different from the ones, I guess, in some sense, asked in ecology and closer to, but again, different from the one asked in population genetics. So the motivation is very, I'm giving just a very bare bones explanation, is that clearly in a cell, things are localized in various places. Also reactions where they happen may be localized within the nucleus, within the cytoplasm. So there's many, I think I don't need to argue to this audience that there are many spatial factors that go in that, go against the idea of modeling everything as if it's well stirred and everything happens at one rate throughout the cell. The part that sort of motivates me to get into this is the stochastic factors that arise from low molecular counts, the fact that reactions in cells are inherently stochastic, and some molecules. So this is the part where I first got interested in this to look at sort of multi-scale results. Is that some molecular types are quite abundant, let's say different orders of magnitude, and others are only in a few molecules. So sort of any classical limiting or classical approximations would not necessarily work in this kind of context. So I took a look at what kind of spatial models for intracellular reactions have been sort of looked at or simulated from. And so, of course, classically there are reaction diffusion PDEs, which are spatial and deterministic models, which are not satisfactory for a number of reasons in intracellular contexts. Then there are spatial and stochastic models. So you can go to single molecule detail, right? Sort of model every individual molecule of every individual chemical species type that you want, model its movement. That obviously is simulation-wise very intensive and stochastically a complex thing to sort of look at. And then, of course, a lot of people look at compartment models, especially for simulation purposes. And the compartment model is essentially, in some parts of the literature, it's called reaction diffusion master equation. So essentially, it's kind of well exemplified in this graph that I stole from a very nice survey by Smith and Grima in the book in math biology. So essentially, you think, okay, things are not necessarily very well stirred because it would take lots of molecules are very far apart from each other, and if the lifetime of molecules is shorter than the distance in the cell, then you can't really argue that the reaction is going to happen before the death of this molecule. So the compartment models are done so that you sort of subdivide the space into enough small compartments where you can argue that things are reasonably well stirred. And this notion of how well stirred means, at least in the compartment model context, is that you want this, you want the average lifetime and the diffusion coefficient to sort of match well relative to the scale that you're looking at. Okay, so the notion of, so for example, if you start subdividing too much, which is the picture on the very right, then you essentially like isolate everything to its own compartment, so where are the reactions going on. So compartment models, the idea is that within a compartment you assume things are well stirred, and you write down your set of reactions, and then you allow the molecules to migrate between the compartments. Okay. However, if you isolate them if you make the compartments too, so there's this trade-off between too large a compartment, things won't actually be well stirred. Too small compartments, things won't have anything to react with because they're all isolated in its own compartment. They have to wait to start moving, so there's almost no reactions because you have not many things within a compartment. So kind of for people who actually look at what's the right mesh size, they look at things like the chromatin length of different molecules and try to match that to the approximation that they're doing.
So,
yes.
sorry, a naive idea would be to say we have some sort of random walk for each molecule, and if they're within a certain radius, then we allow the reaction to take
That's
place.
for second.
Oh, but that can be done too. Okay.
Yes.
Okay, good.
Well, that's essentially a single molecule detail.
Yeah, it's okay, right,
Right,
which you
so-
mentioned, but it, okay, got it.
So that's, how would you simulate that? You would have to do it
But
piece by
that's,
piece.
okay.
The compartment idea is to be able to simulate things more
More
efficiently.
efficient, right.
Okay, so obviously now you want to coarse grain things, right? So, or how are these things interconnected? So if you just, so essentially there are various kind of limits that you can take, and limits have to do with either how many particles you have or how fast they move around, right? So if you start from the compartment model, which is the green thing, then if you diffuse very fast, then you essentially are in a well-stirred scenario, so you're losing, the green to yellow is going to lose the spatial component, right? We're going to average over the spatial component. If you have high concentrations, then you're going to average over the reaction stochasticity, so you're going to go to the PDEs, the reaction diffusion equations. And then, of course, if both of those things happen, if you have a high concentration and well stirred, then you're down to rate equations, which is just your CME, right? Your chemical your chemical OD, right, in the whole space. So there's various ways you can go, and of course it's going to depend on what kind of system you have, what's reasonable, what's realistic for the system you have. So that's what I mean by appropriate use models depends on your particular network. However, the thing that's important, right, is to not, well, from my point of view, is not to get rid of all the relevant stochastic factors, right? Especially in these contexts where we have low counts of certain things and high counts of other things, is that sometimes the low count thing is exactly the element that triggers the right, reaction of the right kind of dynamics, and you don't want to, you want to average that, you want to keep the spatial possible stochastic outcomes that can happen, and stochastic, events can lead to very different phenomena than if you disregard. So sort of my first dabble into this was a bit ago. Feels like a long time ago, probably 5-6 years ago. Let's start with a compartment model framework, and let's see, because those are things that people are writing simulation models for, and let's see what can we say if I actually enter in this idea of different amounts of molecules, or different types of molecules are there in different abundances. So we wanted to really look at, so in a compartment model framework, including Markov chain dynamics within each compartment. So this X, your Markov process is counting the number of species of certain type within a certain compartment, and then you allow, I'm just writing this in notation that you've seen, fortunately, yesterday in data and stuff. So I'm writing the Markov chain in terms of the Poisson process to drive the reactions, but essentially the thing that I'm interested in is that I'm allowing for the rates of reactions to vary from compartment to compartment, because that might now this allows me to say in certain locations, these reactions may not be allowed at all, etc. Right? So I'm allowing for this heterogeneity not only in where things are literally spatially located, but also which reactions can take place in which locations. Okay, so now I enter in the sort of renormalizing of abundances in different ways. And so I have parameters alpha that kind of tell you, I have some scaling parameter n, and I say alpha is encoding whether I have order 1 of certain molecules or I have order n of other molecules. And to keep things simple, let's say I'm looking at just 2 types of like things that are 2 orders of magnitude in terms of differences. And now, of course, I may have to allow also for the rates of certain reactions to be in different orders of magnitude. So I'm renormalizing everything in some realistic way in terms of the scaling parameter n. And then what will essentially happen within the reaction network is that inherently some renormalized quantities will change slowly over time, but will fluctuate slowly over time, and some quantities will change drastically over time. This is all relative to their order of mass, right? So even if something's there in order n, and every reaction is giving me just taking away plus, putting 1 or taking away 1, that's a small change that I consider slow. Whereas if something's there in order 1 for order 1 amounts, and I'm changing it by that amount, that's considered fast. So in some sense, I have this split into fast and slow evolving species types. And the classical example is the mechanism of kinetics, where if you have a reasonably larger amount of substrate and enzymes, then you kind of get this fast sort of binding and unbinding. And so the forward and back rates will be fast. And then the substrate and the product will be slowly changing things, and the enzyme, whether it's free or bound, will be fast changing.
So your N is a concentration of substrate, or it's
in this context, yeah, it's something that's on the order of the substrate, concentration of substrate, yeah.
So you both substrate and enzyme that's at order of N, or?
No, enzyme is order 1.
Yeah, where's kappa 2 then? Large, if enzyme is order 1.
Then your K minus.
You sort of assume that the unbinding is fast, right? So you have.
But if your enzyme is only 1, then your K minus 1 should not be proportional to order of N. Your K1 should be.
It's the rate of it. You assume that it's an unstable complex, so that the parameter. I'm not putting in what I say here is not putting in the scaling that comes from the amounts. It's the intrinsic reaction constant that I'm assuming that's faster.
Yeah, but what I'm saying, if you assume enzyme only 1, the ES is only 1 of them. So going
Yes.
back, the association is only 1. So it
But
should
what I'm
be-
saying is the rate of dissociation. You assume the rate of dissociation is
It's a particular timescale separation. It's not actually
Yes.
the one that's used in the classical Michaelis mental.
I should not use the word.
I'm not sure that that's.
No, I think the issue is
So
what you're saying.
isn't there a context in which I can say it's an unstable complex?
Yeah,
and it will
absolutely, but-
unbind fast,
but
faster
in-
than binding?
But it's usually not necessarily through that timescale separation. I mean, there are many that you can use, but that's one of them.
Okay. So, all right,
it's
so,
binding on binding is very fast.
Yeah.
And the second.
Catalysis.
again, I apologize
So
for
that
my
was
lack
the
of
idea binding
lack
on
of
binding.
chemical knowledge.
So it's nothing to do with
It's a thing that
concentration.
fits my
Scale.
model and gives me
Yeah,
gives
I
me
understand
the
that.
Michaelis-Menten kinetics in the end, surprisingly. So what I'm going to now look at is supposed to allowing the molecules to move different speeds also, because maybe the enzymes don't move very much, the substrates don't move. I'm at this point allowed, that was just an example. In general, I have any kind of set of chemical reactions. I can allow, using these kappas being 0 or non-zero, I can allow any kind of set of chemical reactions to happen in the different orbits. Now I'm going to allow the species to move at different speeds. So here, for simplicity, I'm thinking of one slow species and one fast species, just for the notation, that you can allow each species to have its own sort of speed, which you interpret in terms of the scaling parameter n. And so my question that I was interested in is, how does this speed of a fast evolving species, whether it, so I have a time scale, I'm assuming I have a time scale separation in the reactions, right? That's the first thing I assume, and I assume that this time scale separation, let's just set the scaling parameter n so that the time scale separation is, or I'll do a time change so that the time scale separation is n to the 1, right? So it's order n. So something fluctuates very fast at order, n fluctuations per unit time, and some things fluctuate slowly, only one change per unit time. Now, how does the movement, whether it's slower or faster than these reactions, how is that going to affect my end dynamics? That's kind of the first question. Also, how do the distributions, where these, because I'm allowing any kind of movement, right? It's not uniform or anything, they may want to go certain places, so I assume that the movement is, has some stationary distribution, then how do these distributions affect, again, my rates, my final dynamics? So I worked on this kind of general setup with a colleague, Lea Popovic from Freiburg in Germany, and we kind of characterized this, the overall stochastic dynamics in terms of, so the thing that we were in the end interested in is, let's say you can't observe what's in what compartment, you're just going to sum up the total number of each species within the whole space. And so we wanted to write down what's the dynamics in the end of the sum total. Okay. So we characterized it depending on all of these, all of these 4 different cases. I have different speeds. One is up, one is down, 4 combinations, and you have sort of, you think, oh, I'm going to get 4 different answers, right, in the end for how things end up. And what we observed in the end, so I'm not giving you, God forbid I should put the notation for the theorem, etc. But there are 2 kind of consequences that we noticed, which actually intuitively make sense. But in our mathematical results, they fall out as corollaries. You can rigorously prove them that those species that are in high abundance, their speed does not really matter, whether it's faster or slower than the reaction dynamics. So how fast they move is irrelevant, and in some sense, the only thing that enters into the final calculation is where they end up, like what's their stationary distribution. And in some sense, intuitively that makes sense. I don't have to put money on it, or a lot of money on it. Makes sense because in some sense, they're abundant. There are a lot of them. It doesn't matter how fast they move. Probably in various portions, with good chances, there are species like this in all portions of the space, right? So it doesn't actually matter if they're going to well mix or not, because there are just so many of them relative. The other things that we have observed is that for the fast reacting species, so now in it's the ones that fluctuate, whose abundances fluctuate relative to their amounts very fast, the dynamics does not depend on what their distribution is, where they want to end up in space. The final dynamics depends only on whether they move faster than their reaction fluctuation, the reactions they're getting to, or they move slower than their reaction fluctuation. Again, this makes sense, because who cares where they end up? Since they fluctuate so fast in a reaction, they don't have the time. They're going to disappear before they have the time to actually reach wherever it is they're going, right? And their speed matters because if they fluctuate fast, so if they, in some sense, if their speed is faster than the reaction fluctuations, then you can kind of think of them as well stirred, right? Because they'll get somewhere. And if they move slower, then no, then you really kind of have to react first, and the reactions are going to, essentially what happens is that the reactions are going to change their amounts more than any movement. So that's kind of the take-home message for that. And there's some kind of nice little corollaries about if you have a mass action system, simply because the mass action rates do work well with this kind of, if you have a finite number of compartments, then essentially you can kind of, you should have a multinomial distribution as your stationary distribution for where the species end up, and the multinomial spatial distribution goes very well, averages very well with mass action rates. So you can actually observe that if the fast-acting things, if the fast-acting species move fast, then in fact it's as if you're in a well-stirred system, because the rates that come out are exactly as if you're in a homogeneous environment, except that you maybe average the constants with respect to something. However, if the fast-acting things are slower, then you really get a different form out. So let me just give the example in the Michaelis-Menten case. So if enzymes move fast, then it's as if you have a homogeneous system, except that now your chemical constant rates are there. You have to enter in, this PI should have really been I of S. So it's where the substrate would be. So you look at the distribution of the substrate, and then you average the chemical rates over that. And that's your overall, those are kind of the chemical rates that you will use. You can replace the whole system by a single well-stirred system, but you just replace the chemical rates with some other ones. However, if the enzymes move slowly, the total number of substrate has a different kind of kinetics, right? So this, the undergraduates should know that this ODE is not the same as that ODE over there, because sums and the nonlinearity, unless you have linear rates in the system, these two things are going to be different, right? I mean, the averaging is going to turn out. So in this case, the fast-moving enzymes lead to faster substrate degradation. So this is essentially slower than this, unless your substrate is, unless all of your distributions are 1/d, unless everything. So there were some kind of, I guess I should back up in terms of some kind of things to be learned, which is that the spatial heterogeneity can both increase or decrease the turnover in the species. And the only thing that you can kind of consistently observe is that the level of nonlinearity is reduced if the fast-reacting species also move fast in space. It's somehow like the signal is attenuated because you don't have such sharp responses. If the fast-reacting species are also moving and reacting with everything else, then your signal, if you have some signal response, it will be attenuated, whereas it will be sharpened if you keep them localized, right? So we've applied this to a few other examples in this other paper that we wrote for the Journal of World Society Interface for signaling pathways or something like that, where you really can show that localizing things makes sense because you will get a sharper signal as a result, rather than having things homogeneous or moving too fast. Okay, so now we're on to the second portion, which is, okay, I was not, compartment model great, but that's not really how things happen, and, that's already an approximation of something, and then if I'm going to do that, then I would have to figure out what scale of the compartment. So let's go back to the full, molecular kind of molecule by molecular model. And so then we said, okay, how do we put this in a nice stochastic process that then we can apply sort of model reduction, stochastic reduction tools like stochastic averaging too. And so with a colleague of mine in Paris, we looked at this framework where you encode everything into as a measure value process, because then I can encode, when we first start with everything counts as one molecule, right? Like, so you have little delta masses in different parts of the space, and then exactly this that you suggested, we put in molecules that are within some epsilon distance. You can now put some kind of interaction kernel around it, because further or closer may contribute to the probability that you actually perform the reaction, right? So essentially you think of, I'm going to put point masses to where the molecules of different types are, so I'm going to have to keep track of the measure process for each of the different types, and then I'm going to have this interaction kernel when it comes in. Now, of course, if you're putting that into the stochastic process, the nicest way to sort of encode that is, because that's exactly what I said, there's delta mass of the different metals or the types, the Y is the location, and the nicest way to kind of encode that is assuming we want to model this in a Markovian way. So I guess I did not mention somewhere in the previous slide that, yeah, the movement. Now I'm allowing the molecules to move in any kind of way as long as it's Markovian, right? So they can jump, they can stay put, they can diffuse, but if you're going to use if you want to use some kind of crowding argument, then encode it in a Markovian way, and then exactly. Okay, so what's the point, or what's our goal with this framework? Is that now, let's see if we can do some reverse limit in results for coarse grading by exploiting different timescales, or by exploiting the orders of abundances, etc. So there are all these different kind of scale separations, and different things apply to different different chemical reaction situations. So let's look at a gene regulation example. So you have a gene turning its, they're self-regulating by negative regulation, so it creates mRNA, which then creates protein, and then this protein goes back into the nucleus and says, turn yourself off. I've been made. No need to be, the gene does not need to be on, right? So the localization thing here is that, well, the genes are in the nucleus, right? So they are staying put. The RNA and the proteins can move around, but I guess that's on the next slide. I just wanted to point out that there are two rates that are not just chemical constants, right? They're constrained to be within this B delta, which is the nucleus, and the other one is constrained to be outside of the nucleus. So that's this thing, which is, so the proteins in RNA will diffuse, while the RNA actually needs to move outside in order to make the protein. So the protein translation only happens outside. Okay, so I'm already constraining some reactions and how they can happen. Okay, so what can we do with this? Well, the thing you don't want to do is to say, okay, let's suppose this protein, so what would make it well stirred would be to say, well, the protein is really fast, right? So it can signal, it can switch the gene off very fast, because it's going to go right back as soon as it's being, the RNA creates it in the cytoplasm, but if the protein is really fast, I'm going to diffuse very quickly, I'm going to reach the nucleus back and signal that it should be turned off. So you get some kind of, so essentially all you care about is this probability of, going back into this one, you care about the probability of the protein being within the nucleus, right? The proportion of protein that are within the nucleus, and if the diffusion is fast, then that will happen sort of the only thing you will care about is essentially the total mass of the protein and then the volume of the nucleus relative to the volume of the cytoplasm. So in the limit, you get some non-spatial Markov chain for the amounts of total mass of RNA and protein you can look at, and of course this behaves as you think. It has some kind of, goes to some steady state. You don't see anything particularly interesting. Now, if you don't allow the proteins to go very fast, and you assume that sort of the signaling is happening faster than that, then essentially you have some local regulation or local feedback because it's only the proteins that are close enough to make it into the nucleus to regulate the switching that will guarantee this. So now you can now replace your set of reactions by these sort of reduced system of reactions where you have to compute at each time point what proportion of protein mass is actually within the nucleus, right? And that's now coupled, unfortunately, with the dynamics of the other things. And so you don't unfortunately get, I guess what I'm trying to say is, you don't actually get a Markov chain because you have to know where, in order to compute this, the mass that is going to make it into the nucleus, you actually would need to know where all the proteins are. So you get very different results, and as is probably not surprising to you, what you would see is oscillations in the amount of mRNA, and meaning you're seeing the expression up in the amount of the amount of protein go up, then go down, then go up, and go down, which actually, if you model things deterministically, you would not see them because the oscillations have random, amplitudes. They have random, like what I call these. I'm always hesitant to call these stochastic oscillations, but I don't know. But how is that defined, right? But you can see that it's doing some oscillatory behavior, but there's randomness in terms of there's no fixed period, there's no fixed amplitude, right? So if you were to model this with a deterministic system, the oscillations will attenuate over time, but in the stochastic system, and the oscillations are left due probably because of this delay. The protein needs to actually, once it's created, it needs to make its way back to the nucleus in order to regulate it. So there's some delay, which is causing the oscillations down. There are other kinds of limits within this. This was like a toy system we wanted to play with. You can imagine that the proteins are very abundant. This would give the same limit as if they're kind of moving fast, if they're really, really abundant in order to the switching mechanism. You can kind of enter the burst kind of behavior, taking into account the lifetime of the protein versus the RNA. There are all sorts of things. For me, the exciting thing is that within this new framework, you can, obviously, in the simplest scenario, you can get the reaction diffusion PDE, right? But there are other interesting stochastic limits that come in. So one, if you have different abundances like this, of like gene versus high abundance of protein, what you would get is under all these nice appropriate assumptions, you would get a Markov chain coupled with a PD, right? So the Markov chain is telling you the counts and so it's a spatial Markov chain, right? Because it's a discrete measure value process that's telling you where the changes are in the low abundance things. And then there's a PD that's telling you how the reaction diffusion equation for the things in large abundances are, and the two are coupled in that when the Markov chain changes, then all the PD constants are going to change, right? So you have this kind of triggering of change within how the PD dynamics is. Now, of course, I would like to examine all these things like existence, regularity of these processes, but that's kind of a long-term thing. But my point is that I was pleased that I think there's some new mathematics that's come out of looking at these kinds of systems that I don't think I've noticed. If anyone has seen these things in other contexts, I would welcome to hear that. Thank you for your time.
Questions?
Yes.
For the first half of your talk, can you generalize this further if you have more than 2 timescales?
Yes. It's just, it's, you can have more than some, you just have to kind of do it systematically. You just have to average the right things in the right order.
Another area where a little bit of this, in a different way, appears is if you look at bacterial motion,
Okay.
say E. coli motion, then typically the concentrations inside the cell we take to be continuous variables because Ky and all these guys are very high concentrations, but the switching in the bacterium to decide in which direction it's going to go, it's a Poisson process, counting process, but it is with a rate which actually depends on the continuous variable. So you have the mix between this continuous time, nonlinear deterministic system, which could also be stochastic if there's some external noise affecting the bacteria, but then that's actually controlling the rate of the actual Fokker-Planck equation. So you have this link also between the two.
Great, thank you.
Yeah.
I'll-
Actually, we have a paper to talk about that.
Yes.
There's one context where, in biology, where a lot of measurements are done on molecules that are geared to slides in one way or another, and you can do various kinds of measurements of enzyme parameters using the behavior in the surface. So surface plasma resonance is one example of that. And as far as I know, I haven't looked at these properly, but essentially none of the processes of fitting the measurements to the assumed model take any account of the spatial heterogeneity and the fact that you're actually in the boundary layer of the medium, and almost certainly your diffusive characteristics close to the boundary are very different from they are the bulk of the medium. It will be kind of curious to see if you did a very careful analysis, which sort of at least made minimal assumptions that there's some spatial heterogeneity in this, whether you would get dramatically different estimates for the quantities you're trying to measure. I just don't know the field, but that strikes me as one place where there's data, there are lots of data, and that the assumptions people make to back out the parameters from the data are probably much more simplistic than what you can.
Another source of data is there's this beautiful high-resolution pictures of cells as extremely crowded environments, and people measure things like the time it takes for signaling from a receptor to a gene expression event. There's a lot of data on that, and you have this diffusion in extremely crowded environments. We have quick reactions taking place as well. So that's another big source of data.
Related question to that. So I don't know if you had mentioned it, but what's the effect of the type of diffusion that you have? So if diffusion is super slow or results, or is there an effect?
Well, it really depends. Like I don't have any for this kind of model. I don't have any. I didn't derive any general result. Right. So other than high concentrations or everything well mixed, you're going to get reaction efficiency, right? But yeah, I don't have. It really depends. I think it depends on do you have a multi-scale? Like you have to set up the context that you're interested in, and then say what's the effect of the speed of the diffusion on the final.
Okay, if no further questions, let's thank Lea.
So lunch should be upstairs. Can we come back at 2?
Yes, sir. Great. Extremely clear. Yeah, no, my problem is well, Tom.